The understanding of Listeria monocytogenes in the produce industry began to take shape in the mid-1990s, when positive samples were first detected in salads and whole commodities. These early findings exposed important gaps in facility design and sanitation practices. A pivotal moment occurred in the 2011 Rocky Ford melon outbreak. This outbreak drew national attention to the danger of environmental pathogens and intensified research into how they move through production systems. Since then, scientific priorities are focused on pathogen transfer, survival in diverse environments, biofilm formation, and the search for more effective cleaning and mitigation strategies. Over the past decade, the produce industry has recognized the critical need to control Listeria monocytogenes and related environmental pathogens. The Center for Produce Safety has funded research that demonstrates that facility and equipment design combined with soft or porous construction materials can create niches where Listeria can persist and evade sanitation efforts. A consistent, comprehensive cleaning and sanitation strategy enforced by a risk-based environmental monitoring program or EMP is essential to verify that cleaning processes and procedures are truly effective. Research confirms that Listeria species including L. monocytogenes are ubiquitous in natural, non-agricultural environments across the United States. Both Listeria and Salmonella are found throughout the fresh produce supply chain from field all the way to retail. The best defense is aggressive cleaning and sanitation, supported by an EMP that performs daily, risk-based testing to confirm the effectiveness of cleaning processes. Meanwhile, new technologies and tools continue to emerge to improve Listeria control. Researchers analyzed over 1,000 soil samples from natural, non-agricultural sites to create a national map of Listeria species. Key findings included 31% of soils contain Listeria species and 12% contain L. monocytogenes. The diversity of isolates was high, including 14 known species and 5 potential new ones. L. monocytogenes were more prevalent along the US East Coast, with decreasing prevalence moving westwards. And whole genome sequencing revealed 3 major lineages of L. monocytogenes, some genetically close to clinical strains, though genetic similarity does not confirm direct transmission. This map now serves as a key reference for outbreak tracking and risk assessment. Show that aggressive cleaning and extended sanitizer contact times are critical for pathogen removal. On clean stainless steel, effective sanitization can occur within 30 minutes, but dirty or rough surfaces require longer exposure. Field research demonstrates that harvest equipment must be cleaned and sanitized at least daily and often mid-shift to prevent pathogen transfer from field to produce. Packing houses are particularly vulnerable. Pathogens are frequently found on floors, drains, conveyor belts, and equipment surfaces, with higher prevalence during rainy seasons when moisture collects. Timing is everything. Cleaning and sanitation should occur as soon as possible after operations conclude to prevent biofilm formation. Investigations in peach packing houses revealed that some equipment had higher bacterial counts after overnight cleaning, indicating that standard routines were not sufficient. Biofilms formed overnight on rough surfaces, allowing Listeria to survive more than 25 minutes in 200 parts per million sodium hypochlorite. End of season and startup periods require extra care because Listeria and Salmonella can persist on equipment between production cycles. Even dry packing environments present high risks. Salmonella and Enterococcus faecium have survived up to 12 weeks on dry surfaces, and Salmonella was detected on 10% of onion packing equipment samples. Operations demand seek and destroy strategies to remove hidden contamination. Listeria in soil or equipment residues is particularly difficult to inactivate, with transfer to produce occurring even when sanitizers are applied at 250 parts per million. Biofilms create a protective layer that make Listeria cells over 10 times more resistant to antimicrobials than free cells. Quaternary ammonium compounds and chlorine are the most effective sanitizers, but only when used at label concentrations. Quartz should be maintained at 300 to 400 parts per million, and if concentrations fall, Listeria can survive even at 18 parts per million. Contamination risks continue beyond the farm and packing house. A study of 18 distribution centers detected Listeria in 12 facilities, with positives in shipping and receiving areas, cleaning equipment, storage rooms, and floors. Porous surfaces such as epoxy coatings and fabrics, as well as areas where water pooled, were key harboring sites. Retail settings also present risk. Foam cushions and cantaloupe displays showed 18% contamination with Listeria, demonstrating the need for strict, documented cleaning at retail to protect consumers. To enhance cleaning beyond traditional chemicals, researchers are evaluating next-generation technologies. Promising approaches include Dorson coatings, which reduce Listeria biofilm formation on legacy equipment, food-grade spray on antimicrobial coatings, capable of achieving 4-log reductions of E. coli and Listeria monocytogenes on contact. Bacteriophage cocktails, which specifically target Listeria or Salmonella, and finally, lead light treatment and superheated steam, which provide chemical-free sanitation. Center for Produce Safety has explored a range of promising tools to control Listeria and Salmonella. Now, while most of these tools remain at the laboratory stage, the path towards real-world use is becoming clearer. Bridging from lab to commercial application will require dedicated research and development, careful prototype testing, and cross-disciplinary collaboration among microbiologists, engineers, and industry partners. Adoption will ultimately depend on a cost-benefit analysis, considering the ability to reduce downtime, minimize labor costs, and avoid the economic risk of product recalls. For industry operators, the immediate opportunity is to leverage CPS research to strengthen cleaning and sanitation programs. Facilities should evaluate the value of new interventions while addressing specific contamination risks. Special attention is needed for legacy equipment, which may not be designed for easy cleaning, and for new electronic equipment that cannot tolerate heavy water use. Where conventional cleaning is difficult, alternative approaches should be explored and integrated into sanitation plans. For researchers, the next step is to expand the existing knowledge base by building on CPS findings. Progress depends on interdisciplinary teams that combine microbiology, manufacturing, engineering, and even finance to translate concepts into commercial practice. Key tasks include validating new sanitation technologies in real-world settings and developing deeper industry insights through collaboration. Another important research need is the development of qualified surrogates, collections of well-characterized Listeria monocytogen strains that can safely mimic the pathogen in testing. These surrogates will allow facilities to conduct real-world studies in produce operations, verify the effectiveness of new cleaning and sanitation products, and identify contamination points and operational vulnerabilities. Now, despite the promise of emerging technologies, the fundamental message remains unchanged. Aggressive cleaning and sanitation combined with a risk-based environmental monitoring program are still the most reliable defenses against Listeria monocytogenes and other environmental pathogens across the produce supply chain.